Hi everyone, welcome to the chemistry division. We have 4 presentations in this division, and at the end of each presentation, we'll take questions from the judges and then the audience. You may ask your questions through the chat or use the Zoom raise hand feature and then wait to be called on. Before we begin the presentations, can the judges please briefly introduce themselves and mention their research field?
My name is Lila Romero. I'm a research faculty here in the area of organic chemistry, so I do a lot of developing new chemical methods.
My name's Brian Lindley. I'm an inorganic chemistry faculty member at Baylor. I started a couple years ago, and we're really interested in designing new ligands for first row transition metal coordination chemistry and catalysis.
I'm Tucker. I am in the 4th year of my PhD here. I am a physical chemist or student, and my research is focused on chemical dynamics, specifically in metal ion catalysis.
Okay. Our first presentation is by Mary Kate from the University of Dallas. You may share your screen and begin when you're ready.
Thank you. Good morning. My name is Mary Kate Belinsky, and today I'll be presenting on quantifying substituent effects on common boral groups. So in Orgo 2, we all learned about electron withdrawing and donating groups. Electron withdrawing groups withdraw electron density from the aromatic system, while electron donating groups donate more electron density into the aromatic. But have you ever thought of quantifying how strong the electron withdrawing or donating power is? So Lewis Hammett wondered the same thing in the 1920s. He wanted to assign a value for each substituent to tell you exactly how strong it is at withdrawing or donating groups. So he came up with something called sigma values. To measure sigma values, what he did was he started with benzoic acid, which is shown on the left, and he added various substituents. And when he did this, he also measured the change in pKa. So he did a titration of benzoic acid and got a pKa of 4.19, and he then would do a titration on the substituted benzoic acid. And whatever the difference is between those two pKa's, he assigned to be the sigma value. So in this case, the pKa decreased from adding this ammonium group. It went from 4.19 to 3.37. So since it became more acidic, that means that the reaction went farther. More of the molecules became ionized. So this was an activating group. The ammonium group is an electron withdrawing group. So what it does is it pulls electron density away from that negative oxygen, and that stabilizes the molecule. And that would be the case with each of these electron withdrawing groups that we see on the top right. If we added an electron donating group, this would push more electrons into that system and destabilize the molecule. So the pKa would increase. This would produce a negative sigma value and deactivate the reaction. So what Hammett did was he took many different substituents, added them to benzoic acid, and came up with these sigma values. And he created a huge table. And scientists since the 1920s have been working on adding to this table. But one thing that's missing from this table is boral substituents. They're not found anywhere on this table. So my aim during my research was to add more sigma values for common boral substituents. It's important to study boron and to understand the properties better because it is highly used in industry and medicinal chemistry. So on the top here, we see this is the Suzuki Crest coupling reaction, where an organoboron compound is used as the starting material, and it creates new carbon-carbon bonds. And these are often used in pharmaceutical drug development. It's the most commonly used reaction in this field because it's non-toxic and it is stable and easy to do. So that's one highly applied area of boron research. And then another is boron is found in many other drugs like anti-cancer agents, including Velcade. So knowing more about the properties, the physical properties of boral substituents could be very important in these fields of research. So in my research, I focused on these common boral groups. I would either synthesize them, attach these groups to a benzoic acid molecule, or some of these can be bought because they're commercially available. So when I was doing my research, the first step was to synthesize or buy the compounds. The next was to run titrations to determine the pKa. And then lastly, I could use that pKa value to find the sigma value by comparing it to the pKa of benzoic acid. I encountered a lot of complications when working with boron because, so during titrations, we use KOH as the titrant, a base, and H2O is also involved. So some boral compounds, when they are exposed to a base and H2O, their substituent will change to something that we're no longer interested in. So we need something that's stable in KOH and H2O. Another problem that I encountered was that a byproduct for many of these reactions is water. So if it is exposed to water, because of Le Chatelier's principle, the reaction will reverse and we'll get back to more starting material instead of the product that we're interested in. So the compounds that I started working with in my research were the boronic acids, meta and paraboronic acids. And because they're commercially available, I could simply just buy them and then we could run titrations with them using KOH. But before I could do that, I needed to make sure that they're both stable in KOH so that they're not breaking down while we're doing the titration. So what I did was I used Nmr Spectroscopy to look at the structure of the boronic acids. So at the bottom of the screen, we have the product just dissolved in water, and there's the Nmr spectra, and we can see the shapes of the peak. When we added KOH, there's a definite change in the peaks, which could indicate that boronic acid broke down when we added it. Then I proceeded to add HCl, which caused the peaks to revert to what it was originally. So that tells us that what had happened here was when we added KOH, the proton was replaced with potassium, and adding HCl then caused the neutralization to occur between KOH and HCl. And that means that there really was not a breakdown of the product that we were interested in. This tells us that we can use titrations using KOH without boronic acids breaking down. So we went ahead and did those titrations. And what we were interested in when we were running these was looking at the half equivalence point, since it's at this point where the peak pH is equal to the pKa. So I made sure to collect a lot of data points around the half equivalence point. And this way I was able to determine that the pKa of the meta boronic acid is 4.2 and the pKa of paraboronic acid is 4.1. Now that we know the pKa values, it's easy to calculate the sigma values. So you use the equation that I talked about earlier, where you just take the pKa of benzoic acid, which is known to be 4.19, and you subtract the substituted pKa value. And when I did this, I got negative 0.01 for the meta product and positive 0.09 for the para product. Now notice that For the meta product, the sigma value is very small. So this tells us that there's not, the boreal substituent isn't having a huge impact on the activating or deactivating the benzoic acid ionization. The reason for this is the meta position isn't connected directly to the reaction site of the transition state through resonance. So because there's not that direct interaction like there is with the para product, there won't be as great of a substituent effect. But with the para product, you do have the reaction site connected to the boreal group through resonance. So there can be a more direct interaction between these groups. So that completed our study of the boronic acids, and we could move on to synthesizing new boreal substituted compounds. So we next tried to synthesize the para BF3K substituted product. And to do this, we added 2 equivalents of KHF2, and we dissolved them in a 20-to-1 mixture of methanol and water. We allowed this to react for 30 minutes and then used rotary evaporation to remove the solvents. And then what we did was we took the proton NMR, we analyzed it, and we noticed that there's a 1-to-1 integration ratio between the 2 doublets. So that told us that there was a complete conversion to the product, to the para BF3K product that we wanted. But one thing that we did notice was that the actual yield was greater than the theoretical yield. And this meant that there must be a byproduct forming. We did a lot of literature searches, and we found that one of the byproducts, the only byproduct of this reaction is KF, a salt. And this is what was making up that extra mass that was in the product. To be able to run the titration, though, we need a pure product. We can't have the KF in there. So the next step here was to figure out how we can remove the KF. The first thing we thought of is maybe we can use less KHF2, and this would mean that there's less production of the KF if we use less KHF2 starting material. But when we did this, when we used any less than 2 equivalents, instead of having the two clean doublets, we got we didn't have a 1-to-1 integration ratio anymore. So there was not a full conversion to our product. And this told us that we do need to have at least 2 equivalents of KHF2 in there for a full conversion to occur. So the next thing we tried was boiling acetone filtrations in order to remove the KF product. So we took our BF3K product and we poured boiling acetone over it. And because KF is less soluble in the acetone than the BF3K product, the KF would get trapped in the filter paper, and the BF3K would go through the filter paper when we're doing vacuum filtration. So when we did multiple filtrations using the boiling acetone, we then examined the fluorine NMR and saw that the KF peak significantly decreased after just 1 filtration. At the same time, the BF3K fluorine peak stayed the same. So the BF3K product was still present and it hadn't broken down. So this seemed like a good method for purifying our product and removing the KF. The only problem was we get a really low yield when we do this. So one way we can fix this is by using, so this proton NMR also shows that our BF3K product didn't break down when we added the boiling filtration. So to fix the problem of the low yield, though, we can use a Soxhlet extractor, which recycles the boiling acetone through the product over and over again until there's a significant enough amount of product so that we can use this and run titrations in order to find the pKa of this compound. So in future experiments, we're going to continue to synthesize boral compounds, run titrations to determine their pKa, and calculate sigma values that we can add to Hammett's tables of sigma values. Thank you very much for listening to my presentation, and I can take any questions.
Judges, did you have any questions first?
Hi, yes. Great. So I did have a question with regard to the challenges that you laid out with calculating yields accurately when having to account for some of these salts that are forming. Have you tried looking into doing perhaps using an internal NMR standard to calculate the yield just using those really nice NMR spectra that you're collecting?
Let's see, so yeah, over here, we know that the KF product is forming. So we've tried a lot of different methods to try to remove the BF3K product. We didn't think of using an internalized standard like that, but one other method that we're working on that may lead to an improved yield also is by using recrystallization with dichloromethane, because KF is not as soluble in dichloromethane than BF3K, so that might be another option for doing this.
Awesome, thanks.
Okay. Thank you, Mary Kate, for presenting. Our next presentation is by Daphne from Southern Methodist University. Before we begin, judges, do you need more time writing your feedback? You're good? Okay. So Daphne, you can start presenting when you're ready. Is by Nam from Texas Christian University. You may begin your presentation when you're ready.
All right. Good morning, everyone. My name is Nam Nguyen. I am part of the TCU Green Research Group, and today I am so excited to share my project evaluating the therapeutic efficacy of a small indole-containing tetraaza pyridinophane for treatment of Alzheimer's disease. So to outline my presentation, we will first discuss the role of reactive oxygen species within neurodegenerative pathology to finding therapeutic efficacy, exploring green ligands in terms of the relationship between structure and therapeutic function, outlining the methodology and results used for the study, and we'll wrap up with conclusions, acknowledgements, and we'll have some time for questions. So to first begin, when we consider Alzheimer's disease, which is a neurodegenerative disease, the current literature pinpoints reactive oxygen species, or ROS, as one of the main contributors to the onset and development of AD. Some examples of ROS include hydrogen peroxide, as well as the free oxygen radicals, the hydroxyl and superoxide radical. Now, I want to point out the ROS often gets a bad name, but however, it is a very normal part of metabolism as well as other biological processes. However, when ROS is misregulated, it causes a plethora of biological damage, including even cell death. How then can ROS be misregulated? Well, if we have enzyme malfunction, this can certainly cause ROS misregulation. So for examples, we have enzymes such as catalase, superoxide dimutase, or even xanthine oxidase that regulate ROS to a normal level. And again, if they're misregulated, there's misregulation. Additionally, the misregulation of transition metal ions, such as copper-2, also causes the misregulation of ROS. The mechanism of how this works is something I'm going to go into much further detail later on in my presentation, but for now, just know the misregulation of transition metal ions contributes to extraneous ROS generation. So when we consider treatment of Alzheimer's disease, we need to consider mechanisms that target ROS. This is something that we do within the Green Research Group. We synthesize ligands that target Alzheimer's disease. The two mechanisms of action that we consider against ROS is first quenching ROS. We want our molecules to react to the ROS rather than the ROS reacting with some biologically integral structure. And we can categorize this into two subsequent categories: antioxidant character, which is a general catch-all for any type of reaction with ROS as a whole, as well as radical scavenging reactivity, reacting with the reactive oxygen radicals. Additionally, we want our molecules to be able to bind and stabilize transition metal ions and prevent them from participating in extraneous ROS generation. So to exemplify the ligands within our Green Research Group library, I want to demonstrate two ligands that we have studied in the past, PIN3 on the left and OH PIN3 on the right. Our ligands share many similar characteristics. Probably the most hallmark characteristic is that they're small and pyridine containing. Next, many of our ligands contain multiple aza or nitrogen centers. So for example, both of these ligands are known as tetraaza, having 4 nitrogen centers. This translates to therapeutic function as these aza centers are able to bind and prevent metal ions from participating in ROS generation, something that we have seen and studied in the past. I also want to point out that these molecules are known as pyridinophanes or macrocycles containing the pyridine moiety. We usually add new ligands to our library by adding or modifying a functional group onto the pyridine ring. So for example, OH PIN3 has this hydroxyl moiety on the carbon 4 position of the pyridine ring compared to the proton of PIN3. This translates to therapeutic function. We see that whenever we have the OH PIN3 moiety, we see radical scavenging reactivity, the ability to quench free radicals, which we see with OH PIN3 exclusively in previous studies and not PIN3. It is in my realm of interest within the Green Research Group to evaluate newly synthesized ligands to see if they have therapeutic efficacy as a viable candidate for treating AD. So in this specific presentation, I will be discussing the indole PIN3 ligand, which was synthesized by group member Christoph Potta. We wanted to incorporate this indole moiety, and the reason why this indole moiety is of therapeutic interest in the first place is because it is a powerful antioxidant and radical scavenger, but most importantly, it is permeable to the blood-brain barrier. Our ligands are wonderful antioxidants, however, they actually are quite poor at crossing that blood-brain barrier. The current literature pinpoints the indole moiety as being able to cross the blood-brain barrier across many different molecules. So for example, the ever so popular hormone, melatonin, regulating sleeping and waking cycles, has that indole moiety and is able to cross the blood-brain barrier. So the central question of our study is, how does this structural change attaching this indole moiety to the pyridinophane scaffold affect therapeutic efficacy? So we want to know, does attaching this indole moiety to the macrocycle affect the indole's ability to be a radical scavenger? And then how does this compare to other ligands? Next, we want to know if attaching the indole group to the pyridinophane scaffold affect the macrocycle's ability to stabilize transition metal ions from participating in ROS generation. So to answer our first question, we utilize the DBPH UV-Vis spectroscopic assay. DBPH, or 2,2-diphenyl-1-tetrahydrosyl, is an excellent model for reactive oxygen radicals because it is a free radical and, most importantly, stable in solution. It generates this lovely purple color, and whenever it is quenched and not in a free radical form, it is turned into this yellow color. Most importantly, we can track the concentration of DBPH in solution using UV-Vis spectroscopy. In this study, we utilize the reference radical scavenger, butylated hydroxytoluene, or BHT. If you look at the top right picture, as you move from left to right, there is this conversion, there's this color gradient of purple to yellow, and there's an increase in concentration of BHT as I move from left to right in this zigzag pattern. More BHT, more radical scavenging reactivity, less free DBPH in the free radical form. We can quantify this reactive radical scavenging reactivity with percent radical quench, which takes the absorbance of the negative control and the sample and over the absorbance of the negative control. The negative control here being just DBPH by itself with no radical scavenger. So if you look at the results of the study, the x-axis denotates the concentration range that we use from 1 to 1500 micromolar, and the y-axis represents percent radical quenched. As we look at the blue series, that of the reference radical scavenger, as we increase with concentration, there's increasing percent radical quench, plateauing at around 500 micromolar. If we look at the indole moiety represented by ligand, as with the orange series, we see a mirroring of the same trend. As we increase concentration, there is an increase in percent radical quenched, answering our question that the attachment of the indole moiety to the pyridinophane scaffold does not affect the indole's ability to be a radical scavenger. We wish to compare this then to our previous ligand, OH PIN3. We wanted to extrapolate the values of this graph to a singular value. So we converted the concentration ranges to a logarithmic scale. allowing us to extrapolate an IC50 value. The IC50 value is a concentration for each radical scavenger that is able to quench 50% of DPPH. The lower the IC50 value, the more potent the radical scavenger, as you need less of it to quench 50%. We can see with BHT, there's 155.4, and OH-3 and indole-3 are 362.4 and 386.9 micromolar respectively, allowing us to understand that indole-3 and OH-3 have comparable radical scavenging reactivity. With that question answered, we wish to move on to asking our question, does attaching this indole moiety to the pyridinophane scaffold affect the macrocycle's ability to prevent transition metal ions from participating in ROS generation? How exactly does this work? Well, if you consider the transition metal ion copper-2, when copper-2 is exposed to a reducing agent, such as ascorbic acid, vitamin C, copper-2 is readily reduced to copper-1. In an aerobic environment, copper-1 then reduces oxygen and is readily converted back to copper-2. This redox event of reduction, oxidation, and reduction is known as redox cycling. Subsequent redox cycling in an aerobic environment produces ROS that I highlight here. We can track this process using a fluorophore, in this study, coumarin-3 carboxylic acid, or CCA. CCA is mildly fluorescent. In the presence of hydroxyl radical, it is readily converted to 7-hydroxy-CCA, which is intensely fluorescent. Let me demonstrate what this looks like in an experimental setting. So if you look at this figure, the x-axis represents time and the y-axis represents RFU, or relative fluorescence units, quantifying fluorescence, the intensity of fluorescence. If you look at the blue and orange series, these are the negative controls of our study, which is ascorbic acid and copper-2 only. And you can see that there is no growth of fluorescence over time, the fluorescence only being that of CCA. Conversely, the positive control, which is the ascorbic acid and copper-2 in solution, you can see a growth of fluorescence over time. This is because this redox cycling is taking place, producing ROS and turning CCA into 7-hydroxy-CCA. So we wish to apply this experimental setting to our ligands, which we test in 0.5 and 1 molar equivalents. So let me continue to build this figure. So we fortunately see that the application of the indole ligand does in fact reduce the fluorescence signal. At half equivalents, represented by this yellow circular series, we see that there is a decrease in fluorescence. At 1 equivalence, the decrease in fluorescence is so much that it approximates that of the negative control, answering our question that the attachment of the indole moiety to the pyridinophane scaffold does in fact not affect the macrocycle's ability to stabilize copper-2 oxidation state and preventing ROS generation, signaled by the decrease in fluorescence. We then also compared this to the OH-3 ligand. We see that at 1 equivalence, the two ligands are comparable to each other in terms of how much to decrease the fluorescence signal. At half equivalence, OH-3 is slightly more effective at stabilizing copper-2 oxidation state, denotated by the greater decrease in fluorescence. So in conclusion, we know that the attachment of the indole moiety to the pyridinophane scaffold does not disrupt therapeutic efficacy, warranting the future study of the indole moiety and other ligands. In future studies, we also wish to explore other components of therapeutic efficacy, as well as exploring blood-brain barrier permeability and other small molecules of this assay. I'd like to acknowledge the members of my Green Research Group, especially my PI, Dr. Kayla Green. I acknowledge funding through the National Institute of Health and the TCU John B. Roach Honors College and the TCU Biology and Physics departments for their instrumental use. Thank you so much. At this point, I'd love to take any questions.
I got a question actually on slide 9. You said you extrapolated the IC50 values from that logarithmic curve.
Yes.
What like fitting parameter did you use? Because like I noticed you have really low errors on those IC50s and you don't have a whole lot of data points. So what did you do to extrapolate those values?
This is actually something that my PI helped me do because she had this program that the undergraduates didn't have access to. And essentially, we just took the values that we got from this graph right here, and we extrapolated it to this program. I'm not exactly sure, unfortunately, what fitting she used. She did tell me that we had to remove some of the lower concentrations and the higher concentrations because it contributed to error. And to kind of elaborate on that, usually in previous studies, we've only reported this graph right here that you see within our papers. The calculation of the IC50 value is something that our graduate student, the one who synthesized this molecule, Crest, suggested because we thought it might be better to to find a single value to represent radical scavenging reactivity. We do acknowledge though that this process right here is very error prone. So this is why we hesitate to say this ligand is better than this ligand. We just want to say, we want to find ballpark ranges to say they're comparable because we acknowledge that it is error prone.
Yeah, because like I mean, in order to get an error value, I spend a lot of my time fitting things. It's my entire life is I have to fit. And to get an error that low from data that looks like that would require to use like 2 data points, and at that point, you're just not actually getting any real information from it. Okay.
Right. Thank you for your question.
And Dr. Lindley had a question.
Yeah, really enjoyed your talk.
Thank you.
I just had a question about, I might have missed this, but how confident are you that the indole group stays intact during this study?
Is there a specific part of the study that you're referring to, whether it be the radical scavenging reactivity or the stabilization?
Not a particular part of the study, mostly just, I'm just curious if there's any possibility for this group to basically, or for this compound to degrade and generate the other, the hydroxy species.
Yeah, that's an excellent question. There has been nothing we have found that suggests that the indole compound is susceptible to degradation. One really prevalent biological thing that we have to consider is the possibility of the oxidation of the indole compound. We have noticed that the indole compound is actually quite stable in just regular shelf life in solution, in the fridge. We have noticed that. And we, in our studies, we haven't seen any observed any observations that really suggest that there is this the hydrolysis of the macrocycle to produce the hydroxy species. And with NMR characteristics, we haven't seen that either. But that is a great question. Thank you.
Thank you, Nam, for the wonderful presentation. Well, thank you to all the presenters. You did a wonderful job. Your research was very impactful and very interesting. This concludes the chemistry division.